I hope everyone is ready to start our class. I'm going to explain about immune response, chapter 11, subtopic 11.1, immune response. We are going to define immunity and state the types of immunity. Describe the general structure of antibodies and state the classes based on its structure. Then state roles of lymphoid organs in immunity such as thymus, spleen, tonsil, lymph nodes, bone marrow. Then we are going to explain various types of antigen and antibody interactions. Immunity, it is defined as ability of organism to resist illness or resist infections caused by pathogens or foreign substances. So there is a difference between pathogens and foreign substances. Pathogens are the microorganisms that can cause or brings antigen, so it can cause illness and cause infection to us. But foreign substance might not be a microorganism. It can be the rust, karat, paku karat. It can be any selumbar contohnya kan, yang masuk ke dalam kulit kita. Kaca, glasses, shredded glasses, itu adalah foreign substance. So they are not pathogens. Okay, jelas apa beza antara pathogens and foreign substance. Overview of immunity yang kita belajar ni ada gambar aja ni cedok daripada buku rujukan. So we have innate immunity and we have adaptive immunity. So we are going to talk about adaptive immunity lah. Innate immunity ni memang ada dalam badan kita, includes skin, mucous membrane and any secretion that can help in preventing foreign substances invading our body. Dan ada internal defenses, including the phagocytic cells such as lymphocytes, natural killer cells, and antimicrobial proteins, and any inflammatory responses. So when we are born into this world, we have our innate, memang sedia ada. This usually depends on our genetic factors. Contohnya kulit kita, jenis apa, macam mana, sensitif, kulit sensitif dengan tak sensitif, itu adalah innate, inborn. Then we have acquired immunity. Acquired immunity then will be divided into active and passive. Okay, so for active acquired immunity, this is our own antibodies. Our body produce our own. We produce our own antibody in our body. While passive, we receive antibody from another sources. Mungkin daripada orang lain, dia akan bagi pada kita antibody, kita terima. So that is passive. While active is when our body actively producing our own antibody. For active acquired immunity are divided into naturally acquired and artificially acquired. So natural is when we are exposed to the infectious agent, like in our daily life, when our friends, our close relatives have flu, and then we are exposed to a sick patient with flu, then we will gain the natural active acquired immunity. But for artificial active acquired immunity, the example the best is immunization, the vaccination. Vaksin yang kita ambil yang dia akan trigger badan kita untuk hasilkan antibody untuk melawan sesuatu penyakit tersebut. Itu adalah artificial but then active acquired immunity. For passive, natural passive, maksudnya benda tu kita dapat antibody dari secara pasif datang daripada orang lain. For example, maternal antibodies. So baby receive passive acquired immunity naturally, diterima secara natural. But for artificial antibodies from other sources, maybe another organism. So the antibody comes from another organism and then injected into our body. Kalau maternal tadi natural, dia masuk melalui peredaran darah antara ibu dan anak. So antibody is a very important substance in our immunity. Another name for antibody is immunoglobulin. Immunoglobulin. The abbreviation for immunoglobulin is IG, not Instagram Mesti huruf besar I and then G is small letter. So what is antibody? It is a protein. Mesti ada perkataan protein. Antibody is a type of protein that is secreted by plasma cells that will bind to a particular antigen. Or it is a protein produced by immune system due to the presence of antigen. Ni due tu ataupun ada perkataan lain in response. Maksudnya antibody ni memang akan dihasilkan oleh badan apabila bertindak balas terhadap sesuatu antigen. So what is antigen? Antigen is any substance produced by pathogen. So pathogen, any virus, bacteria or any microorganism that invade our body release any substances that is called antigen. And usually antigen is on the surface of the microorganism itself. Let's look at the structure of antibody. Antibody consists of 4 polypeptide chain. Polypeptide chain tu dia protein kan. So there will be 2 light chain. Light ni sebagai ringan. Bukannya cerah. And 2 heavy chain. Yang heavy ni of course lah yang besar lebih berat daripada yang kecil. This is the light one. This is the heavy one. So you have to be able to identify which one is heavy chain, which one is light chain. Both heavy and light chain are held together by disulfide bridges. They are in the Y shape form. Bentuk memang bentuk Y. Each chain has constant region and variable region. Constant region maksudnya this part is similar in every antibody. Similar in every antibody. Mana dia punya content, kita panggil sequence of the amino acid is similar in every antibody. But what differ them from one another is the variable region. So this part will have a different sequence from amongst different antibody. So the variable region of both heavy and light region, they will combine and forming antigen binding site. So antigen that is produced ataupun that can be found on the surface of the microorganism will be bound on this site. So it is very specific to a specific antigen. Okay, dia akan spesifik lah. Yang ni khas untuk antigen A contohnya. Contoh sebab darah kan, kita darah for blood type B person, they will have antigen B. Blood type A, dia akan ada antigen A. So antigen ni adalah bahan yang dihasilkan oleh sesuatu patogen. Macam kes untuk blood. Jadi darah ada dia punya antigen sendiri dan antibody sendiri. So contoh blood type A, they will produce antigen A sebab ini yang akan menjadi pengenalan dia. But then they will have antibody B. Jadi antibody B ni nanti akan bertindak ke atas blood type B. Sebab tu darah A dengan darah B tak boleh bercampur. Sebab antibody dia bertentangan. Dia akan bertindak balas antara satu sama lain dan agglutinate. Okay, if you cannot understand this concept, you can ask me later. What you have to know is each antibody is specific for particular antigen. Classes of antibody ada 5. 1, 2, 3, 4, 5. Okay. Right. You have students need to remember all of the name or the letters that identify the immunoglobulin. IgG, MADE, GMADE. Okay. So we are going to learn about the importance of each classes. For IgM, it involves primary immune response. We will later learn about what is primary and what is secondary immune response. Jadi kita kena ingat IgM, primary immune response that act as receptor on lymphocyte surface. Okay, lymphocyte ni white blood cell lah, white blood cell. So we can find IgM on the surface of the lymphocyte. So what they do is they will bind many antigen at once because they have more than one binding site kan, dia ada banyak ni. So IgM ni sebab dia adalah gabungan combination of a few antibodies, 1, 2, 3, 4 and 5. So their size is quite large. So they are effective in clumping viruses or bacteria that will promote agglutination. Agglutination ni clumping tu lah. Clumping tu. IgA pula. IgA is a combination of 2 antibodies. It will prevent bacteria and viruses from attaching to mucous membrane. Ni contoh mucous membrane biasa kita dekat hidung kita, dekat esophagus, dekat trachea. Right. Passive immunity on nursing infants. Jadi dia banyak ada dalam mucosal surface. Jadi dia adalah types of mucosal antibody. Jadi A ni IgA ada dalam adalah mucosal antibody. Jadi contohnya adalah passive immunity on nursing infant. Jadi itulah bagusnya bila breastfeeding. Anak selain daripada diterima passive immunity masa dalam kandungan, bila dah lahir dia akan dapat daripada breast milk ibu. Okay, it act as because it is to prevent the IgA are found in the mucosal membranes of our body, especially macam dah hidung, trachea dengan esophagus tadi. So kita boleh tengoklah dia punya fungsi dia first line of defense. Kan, peringkat awal. Jadi dalam air mata pun boleh, saliva dalam mulut pun ada. Okay, IgD act as receptor for antigen on B cell surface. Okay, B cells ni is another type of lymphocyte. Kita lymphocyte ada lymphocyte B and lymphocyte T. So nanti I will explain later the difference between B and T. So IgD receptor for antigen on B cell surface. It involves inactivation on lymphocyte B cell. Jadi tanpa IgD, B cell will not be activated. Jadi akan jadi inactive lah. So kalau B cell inactive, nak menghasilkan another type of immune response, dia akan gagal. So it involves lymphocyte proliferation and differentiation into memory B cells and plasma cells. Jadi bila memory B cell ni melibatkan ingatan ataupun untuk identify the same pathogen in the next encounter. Maksudnya kalau pathogen yang sama masuk kali kedua, Dia sepatutnya ada ingatan dan dia tahu apa antibody spesifik yang boleh dihasilkan. Jadi kalau tak ada memory B cells, they will forget and our body will respond differently. Mungkin tak cekap atau mungkin lambat. Okay. IgE, E for allergic. Allergic ejaan bahasa Melayu lah. Ni kalau awak nak ingat lah. Allergic. So involved in allergic reaction, sebab allergic ni ejaan English di A tapi ni E. Involved in hypersensitive reaction that produces allergies, trigger the release of histamine. Sebab tu bila kita allergic, ada allergic reaction such as runny nose, and then ada bengkak-bengkak kan. Jadi kita kena ambil antihistamine. Antihistamine to counter the production of histamine due to the IgE response. IgG act as markers that stimulate phagocytosis by macrophage. So without IgG, macrophage kita yang macam amoeba yang boleh engulf pathogen, okay, cannot do phagocytosis. They will not know what to do when they came across the pathogen. So IgG helps in secondary immune response and protects against bacteria. Lymphoid organs. Lymphoid organs ni kita belajar dalam lymphatic system. Okay, tonsil, sebab tu kalau batuk kena apa kan, tonsil bengkak, dia akan badan kita akan demam, susah nak makan, dia bengkak kan. Then lymph node, semua lymph node yang ada nampak bintik-bintik ni. And thymus is a very important because this is the organs where the lymphocyte will be matured. Then we have spleen. Spleen is considered as the largest lymphoid organs. We have bone marrow, also important. This is where our white blood cell or blood itself, kita punya blood lah, red and white, produce in bone marrow. Then appendix and Peyer's patches. Okay, bone marrow is the site of stem cells divide, where the stem cell divide and differentiate into lymphocyte. And also site for B cells maturation. Tadi saya bagi tahu lymphocyte ada lymphocyte B and lymphocyte T. So the lymphocyte B produce in the bone marrow and then mature in bone marrow. Sebab tu nama dia B for bone marrow. While lymphocyte T, the lymphocyte will be produced in bone marrow, but then it will be transported to the thymus and mature in thymus. Sebab tu nama dia lymphocyte T. Okay, because lymphocyte T produced in bone marrow, but then it mature in the thymus. The thymus is a small organ in the thoracic cavity of vertebrates, right above our heart. It is a site for T cells maturation. Kena faham both are produced in the bone marrow. But then for lymphocyte T, it will travel to the thymus and mature in the thymus. Thus, it will be called lymphocyte T. Lymphocyte B and lymphocyte T, even though they are involved in the same immune response, but their mode of action is different. Different, cara dia berbeza. Okay, spleen is the largest mass of lymphoid tissues to filter blood by removing the worn out erythrocytes and platelets. So it is a reservoir for lymphocytes and plasma cells. Tonsil is a non-encapsulated masses in mouth or pharynx. Di sini It protects against bacteria and other foreign agents that enter nose and mouth. Kita nganga pun dia akan masuk, kita menguap contohnya, kita makan benda yang kotor. So semua tu ada risiko kemasukan pathogen. Okay, lymph nodes, organ located along a lymph vessel. It will filter the lymph because it contains cells that attack against virus and bacteria. Dia ada lymphocyte, macrophage lah, white blood cell. Okay, antigen, foreign substances that trigger specific immune response. Okay, they are usually in the form of proteins. Antigen biasanya adalah protein atau polysaccharide, and also it can be a glycoprotein. Located on the surface of pathogens. Pathogens can be bacteria, can be virus, or they can exist as free molecules. So antigen involve response from B cells and T cells. Foreign antigen maknanya antigen that is not produced in our body. Kita ni dalam darah kita ada antigen darah kita. Contoh siapa darah O, dia tak ada antigen. Tapi dia ada antibody. Darah A, dia ada antigen A. Tapi dia ada antibody B. Okay. Itu adalah self antigen. Foreign antigen, bacteria comes from bacteria, virus, fungus, parasites. Jadi self antigen kita panggil major histocompatibility complex. Histologi ni biasa berkaitan dengan darah. Kita punya sel lah, sel-sel kita dalam sel badan kita. Compatibility ni compatible, perkataan compatible maknanya penyesuaian. Jadi MHC ni yang menentukan darah kita boleh kenal kita punya darah sendiri ke tidak. Ataupun sel darah putih yang bertindak untuk menelan foreign substance, bila dijumpa dia boleh tahu, identify, okay, this is our cell or this is not our cell. Yang contoh macam yang jadi masalah ialah cancer cells. Cancer cells dia membunuh dia membunuh sel dalam badan sendiri. So cancer cell is cells that identify relatives or kita panggil sebagai sahabat, dia identify sebagai musuh. Jadi itu adalah MHC punya peranan. Okay, antigen. So this is antigen molecule. Dia ada specific binding site. Tengok dia ada kan. Jadi the antibody will bind to the binding site sebab ni compatible. Yang ini akan compatible dengan permukaan yang ini. Jadi when the antibody bind to the antigen binding site, antigen bind to the antigen binding site on the antibody, so we can say that this antigen is being inactivated. So yang ini kita panggil epitope ataupun antigenic determinant. Antigen receptor specificity. Different antibodies can recognize distinct epitopes on the same antigen. Nampak ni? Pada patogen ni, bakteria ni ada beberapa jenis epitope. So the antibody will identify all of the epitopes and then our lymphocyte will produce a specific antibody for that particular antigen ni, epitopes ni. Macam mana antibody badan kita bertindak balas terhadap antigen yang masuk ataupun foreign substance yang masuk dalam badan kita. Ada 3 kaedah. Yang pertama, neutralization. Then we have the opsonization. And then we have activation of complement system and pore formation. Ni memang panjang, memang kena hafal nama dia. Activation of complement system. Complement ni melengkapi and pore formation, pembentukan liang. So for neutralization, dari perkataan neutralize, neutralization means the antibody will attach to the binding sites of virus and prevent the virus to bind to our cells, making them ineffective. Itu neutralization, dia neutralkan. Neutralize the threat. Okay, kalau dalam istilah perang, the threat has been neutralized. So almost the same thing. So kita akan gencat senjata dia. The antibody will be used to inactivate the virus or the bacteria by binding to the virus surface. Then the antibody will not release the pathogen. It will be there until later on a macrophage, another type of our white blood cell, will engulf. Ni yang macam amoeba tu kan, dia akan engulf and digest them. Jadi lumatlah dia contohnya. So they will be disposed of in our body. The second interaction is by opsonization. It happens when antibody bound to antigen on pathogen, then it is readily recognized by macrophage or neutrophils and promote phagocytosis. Okay. Binding of antibodies to antigen on the surface of bacteria promotes phagocytosis. To opsonization. Okay. Yang neutralize, beza dia ialah neutralization ialah proses dia pelekatan ni. Okay, pelekatan antibody kepada epitope tadi. Okay. Manakala opsonization, when the process of phagocytosis, itulah opsonization. Right, faham jadi dia bila terangkan tadi tu sebenarnya dah terangkan dua. Jadi yang inilah yang dikatakan sebagai opsonization. The third interaction is by activation of complement system and pore formation. Okay, first antibodies will bind to the antigen on the surface of pathogen. It will form antigen-antibody complex. And this is the antigen surface. This is the antibody. The purple is the antibody. So the antibody binds to the antigen, forming antigen-antibody complex. Okay, due to the binding ataupun formation of antigen-antibody complex, will activate complement system. Ni complement protein ni. Jadi kat sini dia tak duduk gitu sajalah. When the antibodies are bound to the surface of the antigen, protein will be activated. That is the complement system lah. So what does the protein do? Next is they will form membrane attack complex. So yang besar-besar atas ni adalah membrane attack complex. So this membrane attack complex will form pore. Jadi dia masuk seolah dia seolah menebuk membuat lubang pada membrane cell of the pathogen. Jadi form pores causing water and ions to enter the cell. So the cell will swell, maknanya membengkak, and then lyse. Kita belajar pasal hemolysis kan, macam sel darah merah tu kalau air masuk berlebihan dia akan pecah sebab dia tak ada dinding sel. Alright, so sebab tu complement system, activation of complement system and pore formation. The complement system is the complement protein. The protein will form pore on the surface of plasma membrane of the pathogen. Okay, you boleh baca semula nanti. Tak faham boleh tanya balik. If a child were born without a thymus gland, what cells and functions of the immune system would be deficient? So kita tahu tadi thymus is where the lymphocyte T is produced. So the cells that would be deficient is the lymphocyte T. Right So immune system akan terganggu lah. Sebab nanti kita akan belajar lymphocyte T ni apa fungsi dia. Okay, any question? No
Madam.
Ya?
Kan tadi yang IgA, IgG semua tu kan? Yang tu kan kita punya first line defense tu memang IgA. Tapi kalau contohnya kita tanya macam first defense kat plasma cell, kita akan jawab IgA juga.
Macam mana soalan awak tadi? Tak faham.
Maksudnya kan kita punya first line untuk defense pathogen semua tu kan IgA kan?
IgA ya, yang untuk allergic tadi.
Yang tu
IgA yang ada dalam mucus tadi, okay? First one.
Maksudnya IgA tu just untuk yang kat found kat saliva mucus situ je. Dia tak
Maksudnya
boleh.
bila kita makan, dalam mulut kita ada ilio. Bila kita makan, makanan tu sendiri kadang ada benda tercemar. Jadi kita punya IgA yang ada dalam ilio tu, dia akan hapuskan sedikit sebahagian besar daripada bakteria yang ada dalam makanan dalam mulut. Yang lain mungkin akan terlepas masuk dalam kita punya mulut bila kita kunyah-kunyah-kunyah. Faham? Bila kita telan.
Betul kalau kita punya kulit yang macam ada masalah, ada pathogen masuk, maksudnya antibody lain lah pula datang kan.
Then the next nanti immunoglobulin lain pula ya kan bertindak balas masuk dalam darah yang dah dalam darah apa semua. So IgA banyak pada mucosa, mucous membrane. Dan dalam darah dah benda lain pula. IgM ada, IgE ada. Okay. Alright, sama. Okay. Ada soalan lagi? Good question.
Madam nak tanya. Pasal antigen musnahkan own cell sendiri tu macam mana? Dia sebab apa?
Antigen musnahkan own cell? Antigen tak musnahkan. Antigen ni dia protein, dia tak musnahkan. Dia cuma trigger immune response. Yang musnahkan adalah sel dalam badan kita. Contohnya sel darah putih kita, orang yang kena leukemia. Sel darah putih dia engulf ataupun makan sel darah merah contohnya. Dia attack our own cells dalam badan kita. Tu dah jadi kanser lah. Kalau antigen dia tak. Antigen bukan sel. Antigen adalah bahan yang dihasilkan oleh sel. Protein biasanya. Faham tak?
Faham.
Dia boleh protein, boleh polysaccharide tadi, ya kan? Bahan dia. Tu antigen. Jadi yang memusnahkan sel badan kita sendiri adalah another cell. Biasanya kalau tu adalah cancer cell ke ataupun leukemia sel darah putih. Sepatutnya dia musnahkan patogen kan, tapi dia dah tak kenal diri sel diri. Jadi dia musnahkan sel dalam badan sendiri.